Thank you, Doctor.
Okay. All right. So we have to continue on the third biomolecule, which is nucleic acid. Right, and then we have another two, basically, lipid and membrane. We have discussed on the carbohydrate and amino acid and protein. So this will be the third biomolecule. All right, as usual, I preferred to start a class with a picture. Okay, so I know this today is actually your holiday, special holiday after our new prime minister had been elected. I'm not sure whether where are you now. Some of you said you are in a bus, or are you all of you are in a college now, or still at your hometown, going back this evening, or how? How many of you are in UPM now?
Maybe dia still dalam bas kot Doktor pagi ni baru nak gerak. Ada yang dah hadir sebenarnya seorang.
Okay, because this week apa ni UPM we have a convocation kan.
Ya Doktor.
So okay, back to this photo. Saya dengar Elisa punya suara je kan. How about others? What do you think about this photo? What is the relationship of this photo with our topic today? Any idea? Guys? Ada cadangan? Any suggestion? Guys? Are you there?
Is it untuk human ke?
Macam mana?
Sebab kita kan hari ni belajar pasal nucleotide. So macam every kita punya ni mesti akan ada sel-sel dia, ada DNA dia sendiri. Boleh relate ke Doktor?
Alright, almost there. So basically, when we talk about nucleic acid or specific on DNA, so meaning it is really present, right? Like forever. When you see this skeleton, I'm sure you think that it is very old, isn't it? Old skeleton, might be maybe like 1,000 years ago of a skeleton, right? So meaning we can identify the DNA, right? From, for example, like from the bone, which is from any organism, including human, where we can identify whether this bone come from which ethnic or from which human type, right? Or from how old is the human, based on DNA, to show that the DNA is stable, very stable, not like carbohydrate and protein. It's easily can be destroyed or denatured. Okay. So this is just to show you the link. Okay. Boleh dapat?
Boleh
DNA.
Doktor.
DNA. So what we call it, archaeologists, right? So when they want to identify, right, when they did do some study, so usually they will collect the DNA and study to see the differences between DNA from people from other country, or maybe from a different uh, era, right? So they can see the difference, right? Okay, move on. Okay, back to our today's topic. Okay, So nucleic acid. So I think most of you already know what is nucleic acid. Basically, it has two types, right? DNA and RNA. So you should be able to name, full name, deoxyribonucleic acid. So by the name, you can already imagine, because you have learned carbohydrate, right? So how the deoxyribose looks like, right? And ribonucleic acid, where it has a sugar, ribose in the structure. Okay, DNA and RNA. So what's the main function of nucleic acid? So it's not only to express our genetic information, but it's also have a main roles in storage and transfer of our genetic information. Right. So what's the difference between these two, DNA and RNA? So we know DNA is primarily found in chromosome, basically in our cell nucleus, and some in mitochondria and also chloroplast. So meaning plant also have DNA, where we the repository of our genetic information. And then we have RNA, where it has three types. We call it ribosomal RNA, or short form rRNA. And then we have messenger RNA, or mRNA, and transfer RNA, tRNA, right? So I'm sure you know what's the central dogma, right? Come or start from DNA, and then it will be convert, or it being split into two, become one single strand, we call it RNA. And then from this RNA, where this coding will have to translate into proteins. And later, those proteins will be involved in its function, either for cell structure or storage or enzymes, right? So this is the flow of the biological information from DNA to protein, right? So this is a simple schematic structure, right, to show how DNA, from DNA to protein. So DNA can be replicated, right? So first it has to be opened up, right, from double-stranded, right? You can see on the top here, through enzyme, right, it will be break up, right, open up the strand, and then it will be complement through transcription, where it will synthesize mRNA, the complement to the DNA sequence, right, from our mRNA, which is located inside the DNA, and then it will go out from the nucleus into the cytoplasm, right? So where it will attach to the ribosome, okay, to translate into protein. Okay, and then below is the structure and function of protein, which you have learned previously on protein, where protein will either give you energy, can use to synthesis and break down other molecules, or storage, right? Or maybe transport, or for muscle contraction, and also cellular communication. Okay? So basically, DNA will be translated to protein, not to carbohydrate, right? Now we move on. So since this is the first part of your principle biochemistry, so you need to know the structure. So the components, like you learn about carbohydrate and proteins, so you need to know how the structure. So meaning you need to be able to draw nucleic acid. So the monomer, or the monomer of nucleic acid, we call it nucleotides, right? Nucleotides, where it has basically three parts, right? So three parts of the nucleotides. So like carbohydrate, it is a linear polymer, meaning it's being attached with many what we call this monomer or nucleotides, which contain 4 different types depending on the bases, right? And then this nucleotides, as I said, it has three major parts. So first is the heterocyclic compound, right? Which contain the carbon, hydrogen, nitrogen, right? And then the second part is the 5-carbon aldopentose. And the last part of the nucleotide is either 1, 2, or 3 phosphate groups. Okay, so the first part is the heterocyclic, where you will see there is another two types of these nitrogenous bases. Okay, where you know that there is ATGC come from. Okay, so that is the part of the nitrogenous bases. Okay, so we look one by one of the three parts of nucleotides. Okay, so this is by right the skeleton or the structure of nucleotide. So in the middle here is the what we call this, the ribose, right? So 5-carbon ring. So oxygen, and then it has one carbon here, carbon 1, and then carbon 2, OH, right? Carbon 3, OH down here, and then carbon number 5 that will be attached with 1, 2, or 3 phosphate group. So you can see that it's a different here at carbon number 2. So if it's OH, it will be considered as RNA. If it's being oxidized, right, or reduced, it will become DNA. And if you can see here, the position of purine or pyrimidine is the at upper part, right? If you still remember, anything that at carbon number 1, if it's the link at upper part, we call it alpha or beta. Upper part here, anomer.
H or OH, Doctor, sorry.
Here, purine or pyrimidine, if it's the upper part of carbon number 1, is it alpha or beta?
Beta.
Alpha.
Alpha ke beta?
Beta.
Right?
Beta kan, Doctor?
Beta, doctor.
Okay, beta. Right. So the structure should, if it's a complete name, should be beta. Beta deoxyribose or beta ribose nucleic acid, something like that. Right. Full name. Okay, so we look on the nitrogenous bases first, the first part, that giving the nucleotides 4 different types, right? I know that you already know that this DNA and RNA have the same, adenine and guanine. So the structure is considered under purine, okay? And then another 2 is pyrimidine, right? DNA has thymine, or usually we use single word T and cytosine. And you know that RNA, the thymine will be converted into uracil, right? So in the transcription, where the double-stranded DNA synthesize the mRNA, so the thymine will be complement, right? Right, or I mean the uracil will be complement, right? With the guanine, right? Sorry, with the adenine. Okay, so anything that adenine in the DNA, it will be synthesized into uracil in the mRNA or RNA sequence. Okay, so this is the 4 or different type of nitrogenous and the purine and pyrimidine. So we need to look at the structure. What is the difference between purine and pyrimidine? Okay, so this is the structure of the general structure of purine. So you can see purine, it has 2 carbon ring, where 6 carbon ring with having nitrogen at position 1 and 3 attached with 5 carbon ring, 5 ring, right? Where the position of nitrogen at position 7 and 9. So there are 4 nitrogen in purine. And in pyrimidine, there are 2 nitrogen at position 1 and 3. So it's only 1 or we call it hexagon, right? So this is the main structure of the purine and pyrimidine. Okay, and then from the purine, so we have adenine, right? So this is the structure of adenine, where you can see that is a NH2 at position number 6, and then guanine, that is a double bond O for oxygen at position number 6. Okay, so make them different, and then there's NH2 at position number 2 of guanine. So this is adenine and guanine. And purines, there are 3 structures, right? Because of the RNA has a uracil, and you can see that thymine and uracil almost the same, right? They have a carbonyl, double bond O at position 4 and 2, right? And then cytosine. So cytosine is present in both, right? DNA and RNA. So instead of carbonate at position number 4, it has NH2. Okay. So for this topic, so you must be able to memorize these 5 structure of nitrogenous bases. Okay. Similarly, like you learn for carbohydrate and amino acid, right? So there is a pattern that make you can easy to memorize. Okay. Then other this consider the major bases. So in our system, there are other minor bases. So like 2 methyl adenine and 1 methyl guanine, right? That involved in our body system, the purines, and another is 5 methyl cytosine and 5 hydroxy methyl cytosine under the pyrimidines. So you can see there is an additional of methyl in some of the position of the structure depending on where is the methyl. So then we put out the name, right? 5 methyl cytosine or 5 methyl 5 hydroxy methyl cytosine. It's just to share with you, right? So the caffeine that is present in our tea or coffee, right? So the structure is basically look like which one, purine or pyrimidine?
Pyrimidine.
Purine.
Purine.
All right, purine, right? So meaning caffeine is instantly can be absorbed by our nerve and brain, right? Because of the very small size. It is very similar to the purine structure. Okay, so that's why we can addicted to caffeine because of this size, right? It can affect our nerves, especially on the brain. Okay, and then how about this structure? Similar to pyrimidine, right? So
Yes.
if you search about 5-FU, it is actually a new drug that been used to treat cancer. So you may search and found like colon cancer, prostate cancer. It has been utilized in medicine because of this structure can easily absorb by the cancer cell, right? Because it's very small and it can interact with the gene or the DNA of the cancer, so that it will help to destroy or inhibit the progression of the cancer cell. Right? And then move on. Okay, so now you have see there are 5 structures, right, of nitrogenous bases, and then as I said, it has to be attached to the middle structure of a ribose or sugar, 5 carbon sugar. So once this nitrogenous is bind to the sugar, we call it the aldopentoses, we call it nitrogenous base, or we call it, yeah, nitrogenous, means the sugar and base is bind. So we know that the RNA is D-ribose and DNA is 2-deoxyribose. Okay. Because of the loss of one oxygen, it makes the DNA more stable, right, as compared to RNA. So we call it, that's why we call it DNA is more stable, and because of this sugar, it makes these nucleotides soluble in water. Okay, we call it nucleosides. The combination within these two, sugar and the 5 carbon, or the 5 structure of nitrogenous base. Okay. So back to the structure, just to recall. So you have learned how this cyclization occurred from Fischer to Howard projection. Okay. So the D-ribose structure, and then it cyclized into furanose to form D-ribose. Okay. So as I said, name should be, for the complete name is beta D-ribofuranose. Okay. And similarly to the D2-deoxyribose, when it cyclizes, it becomes furanose, form of D2-deoxyribose. Okay, so full name, beta D2-deoxyribofuranose, right? And this is the two structure of the ribose that we link up with the nitrogenous bases. Okay. So based on the structure, I'm sure you know that the left hand side is the sugar that attached in the RNA, and on the right will be the DNA. Okay. So nucleoside. So the nucleoside, so similar like you learn in carbohydrate, the linkages between the structure, right? We call it glycosidic bond, right? So for the nucleoside, so the link between the nitrogenous base and the aldopentose is through N-glycosidic linkages, right? And once it's bind, you can see that the name will be changed. If it's pyrimidine, we call it idine, right? The name at the back. And then if it's bind with purine, we call it osin, okay? Like adenosine, guanosine. Okay? Pyrimidine, cytidine, and uridine. And this structure, there is another new drugs that have been developed, right? Because of the size. So basically, if you search the 3' azidodeoxythymine and these 2' and 3' D-deoxynosine had been used to treat AIDS patients because it can, because it is small and it can enter or can invade into the virus or the what we call this, the cause of this AIDS patient that having the problem. So it can slow down the progress of the patient's disease. Right? Okay, so move on. So this is how it looks like. Okay, so the N-glycosidic bond will be occurred at carbon number 1, right? For this purine, bind with nitrogen number 9. Okay, so this is the purine nucleoside. For the pyrimidine nucleoside, the carbon number 1 of the ribose will be bind with the nitrogen number 1, right, of the pyrimidine. Okay, so move on. So this is the list of the 5 structure, mainly, right, of the nucleoside. For example, cytidine, okay, uridine, adenosine, guanosine, and then there is an additional of this uncommon nucleoside, which is inosine, okay, where it linked with the hypoxanthine. Okay. on. So this is just to show you how these drugs of the nucleoside AZT and DDI looks like. So this represent the pyrimidine and this purine leukocyte structure. Okay, so far so good. You can follow.
Okay,
Yes,
Doctor.
yes,
Can,
Doctor.
Doctor.
Okay, very good. So now we move on to the last part of the nucleotides, right? When we said nucleotides means there is a phosphate that bind to the nucleoside. So the binding is based is through ester linkages of the OH group on the 5 carbon of the pentoses, right? And then bind with the phosphate group. So I'm sure you have heard 5 from AMP, 5 from ADP, and 5 from ATP. Or you just mention it AMP, ADP, or ATP. Okay. So the name comes from adenosine monophosphate or adenosine diphosphate or adenosine triphosphate. Okay. So this is the monomer of the nucleic acid. So I'm sure you know that ATP is play the main role in our energy. So you will learn more about ATP, the function, and also ADP and AMP in your next topic with Dr. Adila. So she will focus more on the metabolism. So you will see a lot of this involvement of ATP, ADP, and AMP later, right? For example, guanosine triphosphate, GTP, you will see it will involve in the synthesis of protein. Uridine triphosphate, you will see it's involved in the carbohydrate metabolism. And cytosine triphosphate, you will see it will involve in the lipid synthesis. Right? Okay, so this is how the ester linkages are formed. The nucleoside, right? The OH that present at carbon number 5 will link with the phosphate group, form a nucleotide. So this is single phosphate. And then it will release water. Right? So this is we call it condensation reaction. Okay? In order to form a new ester linkages. Okay, so and then you can see here, and usually if it's only bind with one phosphate, we call it the phosphate as alpha position, beta, and gamma. Okay, so this is how the nucleotide structure of adenosine 5 monophosphate, adenosine 5 diphosphate, and also adenosine 5 triphosphate. Right? And then from this is just a summary to show that the differences between RNA and DNA. From the name, you can also recognize, right? So if it's only AMP, it's basically the structure or the sugar is RNA, okay, or it's ribose. If you see that there is deoxyadenosine or 5D AMP, so many it will be in the form of DNA structure, okay, and so on. All right. So now we look more detail in the nucleic acid. Once we have understand the monomer or the nucleotides of the nucleic acid, so we want to see how these nucleotides bind to each other to form DNA or RNA. Right. So basically, the linkages between different nucleotides is linked where the 5 phosphate group will link to the 3' or the position at carbon number 3 that contain OH group of the next nucleotide. So the linkages is through 3', 5' phospho diester bond. That's why we read the sequence of DNA from 5' to 3'. Okay. And the backbone of DNA and RNA will consist of this deoxyribose or ribose and the phosphate group. backbone mean is like when you see a leather kan, tangga. So the both side where you hold, you pegang kiri kanan tu, that is the backbone of the leather or the DNA. So the middle where you step on the leather is contain the nitrogenous bases A, T, G, C. Okay. So first we look on DNA. So by the name, okay, deoxyribose. So basically DNA must consist of two, right? Two polynucleotide strands. So we call it double helix. So meaning it must have both sides, okay, left and right, or complementary of the bases. Okay, since it has hydrogen bond at position number 2, so it becomes more stable as compared to RNA. And then it has ATGC or ATCOG, right, in their nucleotides. And then based on the study that been done by Watson and Crick, so we can need the 3D structure, right, of the DNA. Okay. Since 1953, they'll be able to come out with the 3D structure. Okay, we using the ball and stick, right? And then you have to remember that guanine will complement with cytosine, and adenine must complement with thymine. So in order for the DNA to form a double helix. Right? All right, so again, the sugar is 2-deoxyribose. And again, it's a makeup of the chromosome in the nucleus, and it's more stable. Okay. Okay, so this is the researcher or the scientist who managed to come out with the 3D structure of the double helix of DNA, where they use this ball and stick, and then they hang it using this red dot stand, right? Then they come out, and then they see that it actually is the double helix structure is twisted, right? Okay. So if you see it's in the paper, so it looks like a straight line or linear. Okay. So again, the guanine and cytosine will complement through 3 hydrogen bonding. You can see here there is interaction. It is not a strong linkages, but it become complement, right, due to this interaction, hydrogen bond. Okay. 3 hydrogen bonding between guanine and cytosine. However, for the thymine and adenine, it's only 2 hydrogen bonding here. And you can see, as I said, the backbone of the what we call this DNA is the component, it comprised of the sugar and the phosphate. See, it binds through a covalent bond, so meaning it's very strong, the covalent bond from the this phosphate group with another nucleotide, right? The carbon, that position number. What's the position just now? 5, right? 3 prime to 5.
Yes.
Okay. So this is how it links to form a double helix structure. So if you draw using a ball structure, so this is how it looks like, right? Very rigid, that it will form a strong DNA structure. Okay. And if you draw like a ribbon, so this is how it looks like, right? So there is a minor groove and the sugar, we call it sugar phosphate backbone, right? And basically it will look like it's a twisted, and it's not like a straight line, it's actually twisted due to the hydrogen bonding in the middle. Okay. And there will be what was the major groove, right? Right. So this is how, and then the size. So between the 2 strand is around 2.0 nanometer. Okay. And then between this sequence is around 3.4 nanometer. And between these two, nucleotide is around 0.34. Just to share with you how the person and quick be able to build the 3D structure. So you see the hydrogen bonding, it doesn't basically bind, right? But it's only a very weak interaction. But due to this weak interaction, they manage to become double-stranded DNA, right? Because of all this interaction in the middle, okay? And then because of the covalent bond between the phosphate and the ribose, become like a backbone of the nucleic acid for DNA structure. Right? We move on to RNA. So RNA is single-stranded, but it comes from the template of or the copy of DNA. It must complement with the original double-stranded DNA. However, the thymine will be converted or will be complement, become uracil in RNA, right? The sugar is ribose, and then the differences between RNA and DNA, it has 3 types. So it has ribosomal RNA, messenger RNA, and transfer RNA, which is all these 3 types will be involved in the transcription and translation of protein. Right? Okay, move on. So you see here, for example, adenine, right? By right, it has to be complemented with thymine. Okay? So this is the double-stranded. So when it becomes just synthesized to mRNA, so mean thymine will complement with adenine in RNA. Then guanine will complement with cytosine, right? And then cytosine with guanine. And however, adenine of the DNA will complement with uracil. So there are no thymine in the sequence of RNA. Right? Just to highlight again or summary of the DNA and RNA, the big difference. So DNA is double-stranded, RNA is single-stranded. The complementary bases, DNA is ATGC, however RNA is AU in GC. The sugar is 2D-deoxyribose in DNA, and in RNA is 2D-ribose. While the nucleic acid for DNA is only 1 type, and RNA there are 3 types. Okay, rRNA and RNA and tRNA. Okay, now we move on to the translation of nucleic acid to amino acid. So we have learned there are 20 amino acids. So just now in the first slide, I mentioned about the central dogma, where DNA will be transcribed and translated into protein. But before that, it has to be linked up to form amino acid, right? So so we know that DNA is double-stranded, so it cannot move out from or to cannot leave the nucleus. So in order for the DNA to leave the nucleus, so that's why it convert or it synthesized mRNA single strand, where it has a what we call it codon, which come from the nucleus. The nitrogenous bases that give a codon table that has a code for specific amino acid. All right, so the methionine, right, of the amino acid that you have learned, the code is AUG. So you see here, AUG is RNA, right? So the methionine in the what we call this synthesis of protein, we consider as the start codon. And then for the UAA, UGA, and UAG will be the stop codon, right, in the protein synthesis. So this is the list. So basically, you don't have to memorize, right, all this codon. But in the future, if you work with DNA sequence, so basically the software will help you to identify if the list, if the position or the sequence is UAA, so meaning it is a stop codon. If it's UAG and UGA, or if it's like AUG in the sequence, so meaning it is the start codon. So you don't have to memorize. So usually we have a software that can help you to determine what is the amino acid that code by the DNA, right? So you can see here there is three nitrogenous that code for one amino acid. For example, we can see valine here. You see, there are 4 codes, right? GUA, GUC, GUG, and GUU. And for the rest, so that's why there are 64 in total. Okay. And then this is to show you again the sequence, how DNA is transcribed into a single mRNA, right? The double strand. So you can look here, for example, these three. We look on the below sequence of DNA. G, G, T, A, G, C, right? Is complement with the single RNA below, right? C, C, A, U. You see, T, A, and U. So this is occurred through an enzyme. We call it DNA polymerase, where it will open up, right, this double-stranded DNA, and then where it will considered to identify at which position, right, they will start the what we call this, the complementary or the synthesis of the mRNA, where it will come from the promoter. So I already shared a video from the YouTube, which can help you to understand further how this transcription and translation occurred in the cell, right? So basically, this transcription and translation is important in the expression of our specific gene in our system. Okay, so there must be a signal for the cell to express certain gene. Okay, so this is how the transcription and translation occurred. Okay, so you can see there is mRNA, and then this is actually the tRNA, right? So this occurred in the ribosome, okay, the ribosome RNA. So this translation, right? So for example, this sequence that is complement in the original of the nucleic acid inside the nucleus, now it is outside, right, in the cytoplasm, in the ribosome, right? So this tRNA will bind, right? Bind and make the sequence or link the sequence of the amino acid until they reach the stop codon, right? The growing, so it will bind to grow the sequence of amino acid, proline, serine, leucine, based on the code, right, of this mRNA. Okay, so basically this is how the transcription and translation occurred. So as I said, this translational will be occurred in the ribosome. So this is basically already outside the cytoplasm. So you will see that the translation will start from the 5 and to the 3 prime. Okay, start codon, and then once they reach the stop codon, so the ribosome subunit will be released, and the complete polypeptide will be released. Can you follow, guys?
Yes, doctor.
All right. So basically, this is will be the last slide. So can you open your Petrobras?
Yes, doctor.
Okay, wait. I stop sharing. So basically, I upload this
Yang
video.
aktiviti, oh video.
Again.
Ingat yang doktor nak cakap yang aktiviti yang satu lagi tu.
Betul. Yes, we are going to do that. So I need a student, anybody who in front of your laptop to share the online activity. Boleh tak? So the transcription and translation you can learn from the YouTube, which is presented by Prof. Dave, right? So I think the animation can help you to understand further about this transcription and translation, right? And then so as usual, I usually like to do some activity after the topic, right? So anybody want to share the online activity? Structure or nucleic acid. Somebody? Boleh tak?
Yang DNA structure model tu ke doktor? Yang bioman bio tu
Betul. So can you share?
Boleh doktor. Sekejap
Elisa Okay, so Elisa lah buat.
Boleh nampak tak doktor?
Boleh.
Okay.
So okay, I hope the rest are ready. So maknanya please help Elisa to play this or to do this activity. Jangan awak seorang pula buat. Okay, ready? Elisa?
Okay doktor.
Alright. So you can
Start.
click the start button. So read out first. Okay. So Elisa, you want to read out?
Boleh je. Okay. The overall shape of DNA is a double helix which looks like a twisted ladder. To the right is a picture of a DNA double helix. Notice that the double helix has two strand sides. So that's the first notes.
Yep.
Okay, so the first question is, what is the name of shape of DNA shown to the right? So the answer is double helix.
Okay. Nice job. Okay.
Next, how many strands are in DNA? So it is two strands. Okay.
Right.
Next, each strand of DNA is made of repeating subunit called nucleotides. Nucleotides are the building blocks of DNA. We will now zoom in and build a nucleotide.
Alright.
So, kita ada satu sugar phosphate group. Lepas tu ada sugar dengan ada nitrogenous base. So that is the first one. Next ada one, kita ke guanine.
Okay.
And then the next one is thymine. And the last one will be cytosine untuk DNA.
Nice.
Okay.
Right,
So what three parts of nucleotide? Kita ada sugar, phosphate dengan nitrogenous base.
Okay.
Okay, so next, in the nucleotide shown below to the right, what is the arrow pointing at? To phosphate.
Betul.
Okay. Next is in the nucleotide shown below to the right, that is sugar, deoxyribose.
Right.
Okay. The last one will be nitrogenous base, tapi dia thymine bukan guanine. Okay. Okay. Ada yang lain nak jawab juga ke?
Sebab awak yang handle. So I hope the rest will try. Okay, can you read out apa tu? Now let's put some.
Now let's put some nucleotide together to build a strand. To the right, you can see one strand of DNA. DNA, however, is double-stranded. Your task is to build the complementary strand that will pair with the strand shown. So, okay, A pair with T, G, C. So, A, G, C. Click dulu.
You have to click where these hydrogen bonds will form. Kena jawab satu-satu.
Okay. So
no, you see, nice. Now choose. You have to click where the linkages, the phosphodiester bond
kat sini kan doktor? Antara Gs.
Itu betul dah tu. So the next tu, bond tu. Tu.
Okay. So, C.
Klik. No,
Oh.
you kena klik dulu kat situ.
Okay. Faham. So, GC. Kat sini. Lepas tu kat. Bukan.
Dekat guanin tu lah. Dekat atas dia, dekat carbon number apa tu, 3 tu. Of,
1, 2,
of the
3. Okay.
phosphate group tu, dekat atas, kat G tu.
Yang ni doktor.
Bukan on the left side. You tak link up the phosphate.
Ni.
Dia, sebelah dia.
Sini
Carbon,
doktor.
yes. Klik.
Okay dah.
Okay.
A, C. Oh,
A dengan apa?
A dengan T, sorry.
Okay. Buat dia
So,
punya hydrogen bond dulu.
okay, lepas tu baru. Okay,
Yes.
faham. G, C. Hydrogen bond and carbon. A, T balik. Hydrogen, carbon.
Okay, you can see, right, there is 2 and 3 hydrogen bonding. There is a okay. I'll show you. Baca sikit.
Well done. You have successfully built a complementary strand of DNA. Notice that your final DNA molecule has 2 strands held together by hydrogen bonds between complementary bases. Also notice that the backbones of each strand are made of sugar, deoxyribose, and phosphate.
Okay.
So let's hold the hydrogen bonds. Sudden response. Okay. Next. So what type of bond joins nucleotides to each other are shown in the diagram. Covalent bond ya doktor? Betul ke?
Yeah.
Okay. Wait. Nervous. Which nucleotide will pair with the nucleotides below to the right? Timing G.
Hey.
Saya salah. I don't know. Okay. Baik doktor tegur. Kalau tidak kosong kau saya lagi. Which nucleotide will pair with the nucleotide shown below G? GC. Okay. Next.
Okay, you score it. Tak payahlah tu.
Stop share ke doktor?
So that will be the activity lah for all of you. It's very simple, right?
Yes.
So yeah, straightforward je. Basically come from the note as well, for practice and to understand. But you have a tutorial as usual, for your for this topic. So I wish to discuss the answer this Friday. Is it possible? Apa ni, on acid. Simple je, tak banyak pun soalan tu. The tutorial. And can you bring your tutorial 3, because saya tak ambil markah dia lagi kan?
Ya doktor.
Minta semua bawa Can you all bring your tutorial 3? I need to collect back because I need to get your mark. Itu you all marking tapi you ambil balik kan?
Sebab nak study.
Can you bring on this Friday?
Yes.
Together with tutorial
Cuma
4.
doktor yang dekat Putra Belas tu kena hantar juga ke ada submission form untuk amino acid tu?
Tak ada. Sebenarnya Putra Belas, for blended learning, I patutnya buat some assignment. So I change. I think most of you will see, right? Ada date kat situ kan? So in order for me to get the blended learning on assignment to achieve lah numbers, so I tukar lah. So as a lecturer, we have to achieve like assignment to student at least three assignment in the Putra Blast. So basically, I have given to you a lot kan. Tapi I put up there as a file. So kalau letak as a file, I cannot claim it as a assignment. So I change it. So no need to submit online. It's just for me lah. As an evidence, I did the assignment for all of you. Maksudnya tak payah submit lah dekat online tu. Just as we do as usual, we discuss the answer, and then you pass it to me, your mark. Macam tu je. Tak apa, you tak payah hantar kat dalam 12.
Okay, doktor.
Boleh
Nanti recording ni nanti doktor boleh bagi dekat saya.
Boleh, sure.
Okay. Thank you, doktor.
Alright. Okay, I hope you have take all your attendance. It will be closed at 11. Okay. So who is attending to this replacement class? Please take your attendance before 11. So if you don't take, so I will consider that you're not attending. Okay, kalau terlupa ke, just WhatsApp me and inform. Boleh eh? Is there anything to ask before we dismiss?
Doktor, hari Rabu ni kita akan proceed dengan lab 4 kan, doktor? So, doktor akan conduct ke?
Betul. So
Okay.
basically this week will be my last week. But since we have, as I said, another 2 topic, lipid and membrane. So I need to meet with you again on Thursday. Is it possible? Thursday night, as usual, to do another replacement class. Boleh tak? To discuss on lipid. Lipid tak discuss lagi kan?
Belum.
Itulah. Boleh tak? Khamis malam.
Kalau Friday doktor free tak? Sebab malam saya takut macam diorang sebenarnya okey je tapi sebab takut dapat sangat 9 sampai 6. So kalau doktor nak proceed Khamis okey je, cuma kalau Friday doktor ada free ke? So kita buat 1-2 hours kelas ke?
Tapi kalau nak buat online lah kot. Malam tu you all ada, hari Khamis tu you all ada kelas.
Sampai pukul 6 je, tapi kalau malam tu kalau online boleh kot doktor. Macam biasa.
Macam biasa saya buat pukul 8-10 kan.
Okey boleh.
You discuss first. Kalau okay nanti I create the link for replacement class untuk discuss on the lipid.
Okey doktor, boleh. Nanti saya confirm dengan doktor.
Alright. Okay guys. So I think that's all for today's lecture.
Okay, doktor. Thank you.
Thank you, doctor.
Alright. See you on Wednesday.
Thank you, doctor.
Bye.